Hi, welcome everyone. I think there's still a few people trickling in, looking at the attendees going up, but I think we're gonna get started just with the introductions as the last few people are joining. So, welcome to today's webinar, "The Physical Lab Meets AI: How Benchling and HighRes Close the Automation Gap." My name is Russell Green, and I'm the Principal Product Marketing Manager at HighRes. Thanks for taking the time to join us today. We've got some really interesting topics to go through. But before we begin, a few housekeeping items. This session will last approximately 45 minutes to an hour. We will have a dedicated Q&A session at the end, but please feel free to submit questions through the Q&A function. We'll monitor the chat as well, just in case some trickle through, but ideally aim for that Q&A button to get those questions over to us, and then we'll try and answer as many as we can at the end of the speaking session. The webinar is being recorded and will be shared with all registered attendees by end of day today. So let's get into it. This webinar is a collaboration between Benchling and HighRes, two companies at the absolute forefront of laboratory orchestration and integrated solutions. And we're really excited to bring you some of the insights from our combined expertise in helping research teams streamline workflows and accelerate discovery through connected systems. There's a few core topics we're going to touch on today. So we're going to hit the challenging connectivity between digital ecosystems and the physical lab. We're going to very specifically talk about how Benchling and HighRes are working together to close the loop on the experimental cycle. You'll see a live demonstration from Crystal and Nari on how our two platforms work together. And then towards the end, we'll do a little bit of a forward look as to what's coming next with some of our AI tools and agents. The two speakers today are Crystal McKinnon from HighRes and Nari Kang from Benchling. Nari is a product leader with over 15 years of experience delivering R&D informatics solutions in life sciences. She is currently serving as the head of automation and analytics at Benchling, where she leads product strategy for instrument connectivity, advanced analytics, and lab automation orchestration, ultimately helping scientists to move from data to decisions faster through AI-driven automation. Crystal is a lab scientist turned product manager at HighRes. She's worked for more than 14 years in drug discovery research, automating early assay design, high throughput screens, in vitro pharmacology assays, and compound management workflows, but recently joined HighRes as a product manager to continue bringing the scientific perspective into the solutions that we create here at HighRes for our customers. And so without further ado, I will hand over to Crystal to lead us into our presentation today.
Thank you, Ross. That was such a nice introduction. So first of all, it's been a blast working with Nari on this partnership. And so what we're trying to solve by working together here is the connectivity in your lab. And so we know that there is hardware in your lab. We know that people have very complicated software ecosystems that they've been working towards. And, day by day, this is getting more and more complicated. So what we're trying to do is build a connector that is going to help both close the gap between the hardware and the software and your multiple platforms. And we're trying to help you build that AI connectivity piece. So I think everybody has their AI pipelines and machine learning pipelines now, and we're trying to help all of our users be able to efficiently do this without as many clicks. So I'll give you a little bit more detail. Well, before we jump into the detail, actually, for anybody here who has not heard of HighRes before, HighRes for the past 20 plus years has been very focused on lab automation and integration. So we have quite a success story with a lot of wonderful users who have our, robotic integration. So whether that's a full, huge integrated lab, or that's, a couple devices with a robotic arm, or even some devices that are just directly controlled through our software. HighRes has been doing this for a very long time, and we're excited to see you all and chat with you all a little bit more about our current projects. More specifically, Cellario OS, which is our orchestration software, and it sits between your hardware and your data layers. So there are multiple components to Cellario OS. We have direct device control, which means if you have just, standalone devices, like I was saying, you can control those through Cellario OS. We have scheduler, which is the software piece that runs all of your integrated automation systems. We have Cellario OS, which is what I'm showing you right now, which is our orchestration software that sits on top of all of these things. And then we have modules to Cellario OS. We have Request, Workflow, Insight, and Connect. And all of those have different functionality that should enable you to have smooth processing in your lab. I should have mentioned this before. Cellario OS is your API hub. So this is your data layer. This is where you can go for that connectivity for anything that you need to do. We have a very accessible API, and we try to expose as much as we can to make everybody's process smooth from end to end. So continuing a little bit further about orchestration, what we can do in OS is workflow creation. We can do instrument and system scheduling. We can execute those workflows, and we can maintain your data continuity. So that's the piece where we're really working with Benchling today, is that data continuity from end to end. And speaking of that data continuity, I'm going to hand it over to Nari to talk a little bit more about the Benchling side.
Thanks for the great overview, Crystal. It's always exciting to hear and see what HighRes team is always actively building out. And I think this sets up perfectly to what I want to share about Benchling and then why this partnership matters so much. For those of you who are newer to Benchling, let me give you a quick picture where we come from and then where we're headed. Benchling is the fastest growing biotech SaaS company, and over the last decade, we've become a trusted R&D platform for the most of innovative life science organization in the world. We started in the academia and building software that scientists actually really love and wanted to use at their bench. And the scientist first DNA is still the core of everything we do at Benchling. And from there, we expanded into the enterprise, and we raised 400 million more from the top tier investor. And today, I'm very proud to say over 1,300 or more global organizations trust Benchling, from emerging biotechs to large pharmaceutical companies. And we've recognized 6 years running on the Forbes Cloud 100, alongside with the most innovative company in the enterprise software. But what I'm most proud of is the science we are helping to accelerate. We are not just a system of record. We are becoming a strategic platform for how R&D gets done. And the partnership like the one with HighRes are the big part of that story, connecting the physical lab to digital one, so data flows and then science move much faster. And quick overview on how Benchling is bringing scientific data and AI all together to speed up the scientific process. At the heart of what we're going to talk about today is the Benchling automation. This is the layer that captures data from any instrument, orchestrates the lab workflow, deliver the analysis end to end. This is where the HighRes integration, you're going to see more detail, lives in Benchling. And then your work cells are connected, your data flows automatically, and your scientists stay in one platform from the run initiation to result all end to end. And then the automation layer sits on top of this strong data platform foundation. Benchling platform gives the unified data model, an open API, and an admin tooling that makes all of this possible. So every instrument event or even every result, every sample state changes is a structure, traceable, and AI ready from the moment that it's captured. And that clean connected data feed directly into your research and your in vivo studies and your development workflow. So scientists reviewing HighRes work cell run doesn't have to re-enter the result or chase the data across many different systems. It's already in context and linked to the experiment that drove it. And then last but not least, and now we are building the AI throughout the whole entire stack to make the scientists go much faster. So with your lab automation generating high quality structured data, we can actually embed those predictive models directly into scientific workflow. So automating toil or surfacing insights and giving you and our scientists more time to design better molecule and then run more experiments. This is the one main problem statement we see a lot, and then problem that makes all of this platform investment really matter. Because even with the right tool, the industry itself is still moving painfully very slow. 10 years and 2 billion, that's what it takes to move a molecule from discovery to approval. And then most don't even make it. And 90% of molecules fail, and then the one they do are actually very expensive and costly to bring it to the market. And then the big part of why this is happening at the bench every day is scientists are losing, average 20 hours a week to manual work, cleaning the data, not focusing on the science itself. So half of our scientists struggle to access the insights they need to make a quick decision. And they're left generating a ton of data, but it's all fragmented and hard to find and all disconnected from the experiment that's created. And that's the actually fundamental data problem we see, not a lack of data. Generating more data more than ever. But the data coming up from instrument tells only part of the story. The experiment design and then the sample context, analytical output, they live in different places or don't even get captured at all. So before Chris and I talk about how we are about to now show and fix it, let's get specific about what we actually meant when we say data and data problem.
Yeah, thanks, Nari. I think that's a really important issue. So if you look even a few years ago into what people were generating for data, we were talking maybe some raw data files, we were talking some numbers that would typically get thrown over the wall and somebody else would handle them, and then you would go from there. Sometimes this was Excel or a text file. And now we're getting more and more robust with our data, especially with the implementation of AI. So you have your experimental design data, so your plate maps, your sample level information, and the steps that are going to go into this process. You have your metadata, so your device files, your timestamps and your dates for every single thing that's happening in real time. Your raw results, which now a lot of people are getting much more heavily into imaging data, for example. So it's no longer just a series of numbers that we're analyzing. And then your analysis outputs, so your concentrations, your hits, your potential sequences that you're getting as your results. They all need to get combined into one central location, and they all need to be analyzed, and they all need to be understood. And so it's more than just a text file and an Excel file. It is a lot of rich information that we're using back and forth from end to end. And that's why we're trying to build this connector to make it a lot easier to have all of these things in one spot and tracked and traceable. Talking about what a complete data set really looks like. And again, one of the reasons why we're collaborating here. So on the HighRes side, so I mentioned we're very into automation. We have an orchestration layer for your lab. So we have that execution metadata. So our timestamps, our steps, any deviations in the process that you were expecting to happen, our system and instrument logs, so those date and timestamps for the actual times that things happened, and our raw results that are coming from those files. So anything that's associated with your run at the end all gets collected. From the Benchling side, we have your experimental design, your scientific intent, so your plate maps, sample level information, like I was mentioning, your sample lineage, your metadata, and your analysis outputs at the end. So your curve fits, your hits, your concentrations, all of that needs to get combined into one spot. And so that's what we've done here that we're going to be showing you today. So your complete data set where your sample lineage is visible, your experiments are searchable across the entire lifespan of that experiment, and everything, importantly, is going to be AI ready, contextualized, and complete, all in one swift motion. Hopefully swift. So overarchingly, we're trying to close the loop for you. So Nari very efficiently highlighted, the problem where a lot of people are spending their time pushing buttons, passing files, and physically moving their data from place to place. And we're trying to make that process a lot easier with significantly fewer clicks. So if you're designing your experiment in Benchling, we're automatically going to hand it off to HighRes work cells or devices. Everything is going to get executed on the HighRes side. The data retrieval is going to happen on the Benchling side, and then the result capture and documentation is going to happen on the Benchling side. And then you can continue to iterate on that loop to make your really robust process happen. Back to you, Nari.
Before we dive into the live product demo, obviously Chris and I was really hoping to bring you guys all in the actual physical lab to show what our integration looked like end to end, but we are choosing the closest one, second closest to the live. So we're gonna do some fun live demo to show this end to end. Before we dive into this flow, I want to kind of walk you through the 5 steps here, what we're gonna cover, and then you can see individual step here from design, experiment all the way down to capture the data. You can see where Benchling or where HighRes actually play, sometimes individually, sometimes work together end to end. And with that, I'm going to share my screen and then let's dive into live demo. And I'm seeing some questions coming in, but we'll make sure that answer your question towards the end of our session as well. All right, sharing my screen. I might see Crystal's beautiful face first. Perfect. Cool. Crystal, I'm gonna ask you the most common question on the team call. Do you see my screen?
I see it.
Perfect. Yeah, this is the place where actually scientists enters and design their experiment. This is a starting point within Benchling itself. So here's actually the entry where scientists may design their ELISA assay. So starting with some of the plate layout, about how your assay is going to be run, about your samples and your concentration and your role, which one's control and standard and so forth. And then within the same place, you can actually track the digital record of your plate or sample within benching as well. So in this example, I have the actually not a couple 96 well plate that has my protein sample with the concentration capture, and that follows the exact design of the plate map that I have up here. And within the same place where you are designing your experiment, you can actually initiate and then sending this information directly to HighRes. So for the demonstration here, we are kind of showcasing how we envision scientists who designed the experiment within the notebook itself to send the record to HighRes. But you can also imagine how this can be all fully automated by maybe no scientist or human involved, just kicking up from through the API as well. So this workflow you're seeing here right now is about sending those plate from those what you designed, and then optionally just mention that at the end of the work set complete, being able to capture those data all back into the same place where you capture the data, and that's it. So I'm going to actually initiate this run. So the moment I actually kick up the whole workflow, what's going to happen is a couple simple steps. One, taking the information from the experiment where I design my experiment about my ELISA assay, plate map, and then a sample, all those information you need to know. And then I don't know if you just caught the quick update from the task itself. So that information is now directly and automatically flow to the next stage about kicking up the HighRes. So just show you what this actually look like behind the scene. So we actually have the pre-configured data pipeline job here. So just zoom in too. So first, passing the information coming from your experiment, the entry directly. And then second, we have this pre-built out-of-the-box HighRes protocol that can connect from Benchling to HighRes Cellario OS directly. You notice here there's information about plate and experiment just got passed to this protocol. And before I just dive into the OS side, I want to kind of show you how this protocol is configured and how it's flexibly actually connect and then integrate with the OS as well. So this is kind of zoom in view of the what this protocol looks like. And then this protocol is configured, again, reading the information directly from the Cellario OS. So in this case, we actually have the 3 protocol that's reading directly from the OS. And then Chris is going to cover a bit more about how this is configured and how we are reading this information. And then in this case, we actually have a one test instance of the OS right now. But as organization who might have a one-to-many system, you can also choose which system you need to talk to. And then we have this critical information here about the port. This is how we actually configure between 2 systems to send information from Benchling to HighRes or one to the other. So for example, this ELISA protocol, we have actually configured to send the plate information and then alongside with the detailed sample metadata itself. But let's say if I choose different protocol, such as like source to destination, like liquid handler transfer, we have a different information that requires the capture from benching sent to OS itself. So that was a little bit detail about how we configure this out-of-the-box protocol itself. But now I'm gonna jumping back to the OS to see how the request we just made a moment ago is now showing up in the OS itself. Over to you, Crystal.
Thank you. So here we are, and this is actually Benchling's live OS account. So if you take a look, we have an order that was just created May 14th. Nari is on the west coast of the US time zone, so that's exactly the time that we just set it up. So Nari, if you would click on that Psi 798 order ID, please. This is an example of what an order looks like in OS right now, and you can see that the status is reserved and submitted. And you can also see in the bottom left hand, there's an ID, number 924. And that's a numerical ID that's associated with the run on the actual robot itself. So Nari, if you want to hop into Scheduler, and then we'll come back to OS and talk a little bit more. So like I mentioned before, Scheduler is the control software that we use for our integration. So Nari is starting that robotic integration. I mean, this is a virtual system, but then she's also going to see order number 924 here and start it off. Thank you kindly. So now all the devices that we need to run an ELISA assay are running right here. The status is changed to active. And Nari, if you pop back over into OS now, thank you so much for doing all of this clicking for me. And would you mind refreshing the page quickly?
Of course.
We have a new status, so the order is now active, and you can see along the middle that we have file attachments and parameters that are going to get filled out as the order completes. But while we're waiting for that to finish, Nari, would you pop over to the protocols tab? I see it just finished, so fun timing.
Oops.
So, kicked you out apparently. So what I wanted Nari to show in that protocols tab is that on the scheduler side, we have protocols there that are getting run. So in this case, we had that ELISA assay. And here we have those other three options. So those are the other three options that are available in scheduler as well. They're all connected. And as I create new templates for myself on that scheduler system, on our robotic system, those are going to get surfaced in OS and then automatically surfaced in Benchling as well, so that we have a constant stream of connectivity from end to end for any parameters, any files, anything you might want to be running. So Nari, if you quickly go back to order management, I'm sorry, I'm making you move all over the place, and go back into that order, the status is now finished. So if you take a look at that file attachments, we have one, a text file, which is the raw data file that came from our ELISA assay read. And then we have a zip file that has all of the metadata and your raw data files that you could possibly have associated with it. So if we had run 10 plates here, there would be 10 plates plus any metadata and log files that are associated with this run. So now we can head back over into Benchling.
Thanks, Crystal. Let's see how this data, the measurement, as well as the compressed file that has all the details about the system log itself. Beautiful. Looks like data is flowing. Yeah, it's processing right now. So while it's processing, just show you example. So the moment workcell actually complete the job, that it's gonna actually send you the we're gonna actually pick up the data from the measurement and log file automatically. In this case, we are simply just showing the one of the plate measurement that was produced at the end of the ELISA assay run. But this can be also retrieve any other system log or troubleshooting log that that's configured to pick up from the HighRes side itself. And then this is, again, as I mentioned, pre-configured data processing step. So you can do not only getting the data back from the HighRes protocol, you can also use the combination of the pre-configured data processing step, or even scripting to managing your data itself. And then not only that, we also have some sort of pre-configured analysis function, like such as running a regression or running a QC step. And then, as always, the live demo, there's looks like it's a little bit slower than usual.
Every time we do it live.
We had a pass to show the pre-recording, but Chris and I really wanted to make this more real and live. So yeah, here we are.
Oh, here we go.
Actually, while this is going through the series of step-by-step, Crystal, anything else to highlight, or what else can be possible flow from the HighRes side to Benchling?
Yeah, I think really just emphasizing that we're not just doing raw data files, like all of those timestamps, date stamps that are associated with your device file runs and everything can be combined in here to essentially give you all the information that you need. And I am very distracted because I love watching everything move through the pipeline, so I love this part.
Cool. Yeah, if it wasn't obvious, obviously you do not have to create this every single time you run the assay. This is all pre-configured, save as template, so you can run it over again, depending on the protocol and assay you're performing. And then not only processing the data directly coming from HighRes side and then running the analysis, we also have an option to now send all the analyzed data back to the place where you initiate your experiment. So jumping back to the first place where we actually design our experiment of the ELISA run. So here is my plate map design, and then the actual plate and sample information itself. And they can configure to bring the data automatically back to, again, where you design your experiment. This is all highly configurable, so you or scientists can actually configure what data is matter the most and what information should be captured from the HighRes work cell as well. And this is what's possible when software, data, and a work cell like HighRes is working all seamlessly together. And then Benchling makes this automation possible, and then powering by the work cell like the HighRes provide end to end. All right, let's jump back to what more to come beyond here.
Yeah, so I know some people who are in here who have already seen this demo, who are already very excited to work with us, and they want to know what's next and what else we're working on. So a few things that Nari and I have also been discussing as far as making sure that we are enabling as many people as possible with this connector. first thing would be increased protocol complexity. So what we just showed you was an example of an ELISA assay. So that's, a single thread of one to many plates that are going through very step-by-step in a straightforward manner. I'm not saying ELISAs are easy or short, but they are relatively straightforward as far as automating them. So we are working towards making sure that we have more complex cases that are going to accommodate, potential liquid transfers or multiple threads of information going from different sources and destinations so that we can make sure all of that is tracking and working perfectly fine for everybody. Next on our list would be more event-based push and pull of files. So what we demonstrated for you today was very specifically the protocol had completed and Benchling came and pulled all those files. But OS is tracking every single event that happens to every single device or every single step that you're working towards, whether that's manual or, on a standalone device or anything. And Benchling and OS are able to connect to each other and pull that information more on an event by event basis so that you can get more real-time data and you can do, your quick checks to make sure that your assay is looking the way it's supposed to be looking. So that's also on the list. That's what we're looking into.
Crystal, I just saw a few heart emojis sending over the number 2. So yeah, as much as you guys are excited, we are very excited about this next step as well.
Awesome. Oh, that's good news. Thank you for telling me because I did not see them. And then last but not least on the list, we're talking about agent-to-agent communication. So we're trying to enable bi-directional data and information flow in both of our locations. So we know that every single lab is going to operate a little bit differently. On the HighRes side, we have our lab assistant, which is in beta right now, where we're able to essentially query what's going on the Benchling side. So if you have a plate that is at a certain stage of your processing, you need to ask about it, what samples are in here, what information do I need to make sure I'm doing this correctly. If you've run into an error or something, you can just walk up to you won't have to walk up to it, you'll already be in there, to our lab assistant, and you can directly ask it in your own language, what's going on here, what's in my plate, what am I working with? In addition to other stuff, but very specifically for Benchling, we should have this bidirectional communication.
And then when we say bidirectional, we mean really bidirectional. So what Crystal showed the agents from the OS side, being able to actually talk through the Benchling MCP to retrieve information. So that's most useful when you're in the lab, nearby the work cell to communicate. And then same thing for the other side of Benchling as well. We can actually communicate to the OS through the MCP and the agent itself. So this is a couple examples showing how from the Benchling AI side to being able to retrieve the protocol information that sits in the OS, and then being able to not only listing protocol, also being able to kick up the protocol and run more interactively through the agent-to-agent integration. Yeah, this is, as you can see from the image, this is real, not a mock itself. So yeah, definitely love to chat more about your use case, your interest, and how our partnership and integration can help speed up your science.
We showed you everything we wanted to show you today. We told you everything we wanted to tell you. So we're so happy to take questions now. But before you leave today, pretty please, if you could fill out the survey on this QR code to let us know how we did, what you think, or if you have any other feedback. We would love that. And I also would love to say that it's been so wonderful working with Nari and having a partner who's so like-minded and wanting our customers' lives to be better.
Right back at you. I have to say, we work with so many partners, but the very special, unique thing about partnership HighRes has been we are always focusing on our customers' experience. We are always about like prioritizing right experience, and then making our mutual customers successful. So yeah, looking forward to talking to you and then how we can better support your science.
That was amazing. Thank you so much, Crystal and Nari. It's so exciting to see these things being demoed live. This is actually a real integration that's working. And one we're so keen to get out to more and more of our customers. We do have a few questions coming in. Got a few through the Q&A, a few through some other mechanisms. So I'll fire a couple at you and see if we can direct them. I'm sure you both want to answer these. So the first question is Cellario OS required? Can this integration be done directly with Cellario Scheduler via its own API? And maybe I'll direct that to Crystal first, and then Nari, if you want to follow up.
Absolutely. So in this case, we are requiring Cellario OS just because that's a more robust API hub, and that's where we're putting all of our, future thinking efforts into continuing to build our connectors and our partnerships. So right now, yep, the connector is that direct connection, the out-of-the-box productized connection is through Cellario OS. And if you have anything to add, Nari, feel free.
No, I have to say, I know we have customer on the schedule as well, but as a the partner who's been building our integration directly, and then there's been extensive like API and then even coverage availability within the OS itself. And then as your lab actually looking to expand a bit more beyond just not one work cell, like multiple work cell, I do think the OS is the right interface to integrate with. There's so much more I know the HighRes team is investing in OS later as well. So very much looking forward to leverage more of those investment and how we can make more robust integration together.
Amazing, thank you. Another question coming through the Q&A was that for a workflow such as the one being demonstrated, how long would you estimate it takes to implement and test to be production ready? And I'll pick on either of you to answer that one. Maybe we'll start off with Nari this time. And sure, there's two elements to this. So I'm sure you both have a view.
Yeah, Krista, you want to take first, especially from the some of the early initial journey from how a customer may evaluate the work cell as a starting point, and then we can talk about integration layer.
Yeah, absolutely. So I think to answer this question thoroughly, we need to know like from what perspective you're asking. So if you already have systems in place, I don't anticipate this is a terribly long back and forth. If you already have, if you don't have a system, for example, and you need that, and this is something that you're investigating, this could take, months to a year, because that's how long it takes to go back and forth, establish that the integration that you want, and then get the software in place in order to have this connector. But if you're looking more for just software solutions, those are probably a significantly reduced time as far as I know. But Nari, as far as the Benchling side, how long does it typically take for a customer to get established on that end?
Yeah, so the demo use case we share, obviously we have a lot of pre-built configuration ready, and then the connector that we show is actually ready. So obviously we've been actually still iterating with mutual customer to build a more production ready. But in terms of the can I get access now or today or tomorrow to start with, it is ready for you. And then yeah, please do reach out to either of our side and love to show and chat more about that.
That's great. And see another one coming in for Benchling analysis workflow. Are you able to update entity metadata through the custom code? So I imagine this is one directly aimed at you, Nari.
Wow, it sounds like definitely a hard benching customer question right there. Yeah, great question. I think based on questions, the short answer is yes. The what we call like automation designer, the workflow, the interface itself, that's been evolved from our current analysis product itself. And then actually over the last quarter or so, we are bringing more functionality that includes not just running analysis, like being able to bring the data directly from your instrument and then now expand to OS, the user as an example. And then the data processing that includes whether pre-built data processing step or using custom code, and then all the way down to analysis. And then not only that, we are bringing some of the benching actions such as creating entity, updating, or even creating plate or transfer. So as Chris and I are actually building out more robust like protocol step, not just capturing the measurement, also being able to track the plate creation, plate transfer, liquid handling job itself, that action is going to be really critical to track the what's happening in the work cell, reflecting in the data side in benching as well. So long way to say yes, and I'm very excited for our customer to leverage the end-to-end tooling.
Amazing. Going to rattle through because we have some time, so I'm going to keep on rattling through a couple more questions. There's another one that's quite specific here. So do I have to define workflow or protocol twice, both in Benchling and HighRes, or only in Benchling? I think it's kind of an interesting question. Is about which components you have to set up in which bit, and maybe which direction this might go in the future state as well.
Yeah, I can start off on this one. So as far as you defining your workflow, you're going to do all of the steps of your experiment, the physical steps, that's all happening on the HighRes side. And then the actual layout of your plates, Nari, you can feel free to chime in a little bit more, but that's going to be on the Benchling side. So you won't be doing anything twice. So what we were showing was that those scientific steps, those are going to be automatically imported into the Benchling side through that drop down. So you can just select those as soon as you've established those on the HighRes side. But Nari, on the Benchling side?
Yeah, actually what Crystal just outlined here, you do not have to define the protocol twice. And that's actually one of the main problem we were out to solve together. So we had a lot of we have a few mutual customer who use both HighRes and Benchling. And then what we saw from their current state was just managing protocol information in both places, define in the OS or define the HighRes first, and then do the same exact action again within Benchling. And then as you science or as a protocol evolves, you have to reflect in both places. And then that was very painful to manage, and then that kind of hindered some speed that you wanted to go. So when it comes to define scientific protocol that execute and interact with the physical work cell, that sits in the OS. And then Benchling is directly pulling the information. So you have only a single source of truth, one place to manage. And then when it comes to managing your scientific experiment metadata, including your sample, that sits in Benchling that can talk directly to OS as well. So again, no more duplicate data entry or duplicate place to manage your protocol and science. We are very excited about this.
Amazing. I think I see one more question in the Q&A, and there might be one in the chat that I might have missed as well. So we'll try and hammer these two questions through as well. So that one here, hi Nari and Crystal, great job on the demo integration. That's great. You showed how the scientific port parameters are synced with Benchling and can be defined in the automation node. Can you comment on other scientific protocol parameters? Is there a similar synchronization in Automation Designer to configure attributes or values? It sounds quite specific to someone maybe using the platform already, so maybe I'll direct that at Crystal first of all.
Yeah, I mean, short answer, not that short of an answer, is that that's the hope. So that's what we're moving towards. So some of those next steps that we're working on is making sure that that is continuing to be something we can synchronize. So Nari showed how, we do change the scientific protocol, the protocol that we're using. It updated some of those ports. So that's the hope. That's what we're going to keep working on. And that's why we need to add more and more complex cases in order to test that relationship and make sure that that's working fine. But that's the dream right there. Did you have anything else to add, Nari?
Yeah. As you saw, like we've been working towards making more configurable as much as it can. That's why it's like, yeah, engaging more with our mutual customer become more critical. Because I think we made a lot, starting from where we actually initially started back in October, Crystal, to now, it's like we are giving more options so scientists can drive those configuration. That's the first step we've been focused. But next is like, we know there's a most commonly used protocol cross between the two systems, starting with ELISA, just one of the analytical example, but we're going to build up more templatized liquid handling steps or plate to plate or more of those commonly used protocol step as an out of the box template. So you can do active the setup to initiate an active use as quick as possible. That's exactly the direction where we're going.
Fab. And I've got one last question in the chat, and then I might close off with one more for myself. So in regards to automation of analysis, what does the development of automating the analysis data look like? Is this feature for more standardized methods, or is it fairly easy to adjust the analytics process in the workflow experiment to experiment? I think this is more of a Benchling Nari question by the sound of it.
Yeah, great question, by the way. So there's a couple, again, the demo we highlight today is like great example when you have any routine assay. So you have the predetermined process for certain data you're getting, you're always gonna guarantee run those the process end to end. That's the beauty of the using the pre-configured analysis template to kick up the run. And then we also have the another tooling, what we call data science agent. So let's say you have any like one-up assay you're running, and then you simply just want to ask some question, exploratory, those data exploratory on purpose itself. You do not have to go through those heavy like pre-configured template and just run the analysis. You can use our data science analysis agent itself to just get the quick answer itself. So we have all those options depending on degree of the templatization you need for your essay.
And I'll ask one last question here that I got pinged over privately. Just kind of wondering, there's probably a good way to round things off. Like, where are you seeing demand for this right now? So where are we seeing demand for this deeper integration? And where is this partnership between Benchling and HighRes heading in terms of driving greater solutions for these people in the market?
I think for us on the HighRes side, we're seeing this need for deeper integration kind of across the board. So we have customers who are in like, production level labs with more biological samples. We have people who are trying to close their loop for very early stage discovery and screening. It's genuinely a need for that reduced number of clicks, kind of really, really across all of our customers right now, I would say. And so that specific shuttling of, your parameters, your raw data, your metadata back and forth from end to end, and not losing any of the steps in between are really what people are asking for, no matter what their science is. And I don't know if you have a similar situation, Nari.
Yeah. So again, echo to 2 point we covered, like one, make it more configurable. So because we have definitely different types of assay customers are running. Some are templatized, some are not. So make it flexible between integrations. Number one, demand. Number two is the demo we showcased was like the great example of how we envision like scientists in the loop to drive this automation end to end. But there's obviously some a lab who's actually driving more close to like autonomous like process in lab to like machine to machine fully drive. So that's going back to how Chris wrote me earlier. The more event-driven integration is going to be top of our mind, and then where we're going to focus next. So less of the scientists click through some of the steps, but rather truly like data-driven and then event-driven and agent-driven integration is where we see more demands from the larger company, and then where we're going to invest more.
It is such an exciting time. This is such an exciting partnership, and there's so much more to come. I'm sure we're going to be doing another webinar on this in probably a small months because there's so much development going on in this space at the moment. That's been absolutely fantastic content today. Really excited to share all of this. I guess a quick plug from both of our sides. Like, if you want to learn more, then please do get in touch through our respective websites. I realise we haven't got contact details up on this slide just here. Also, there are a number of significant conferences coming up over the next couple of months. We've got SLA SEU, Future Labs Live in Basel, Bio IT World in Boston, and we're at both of these events. And so please come and get in contact with us if you're in the other physical events. And we'll look forward to hearing from you all soon. Thanks so much, Crystal and Nari. And I think we'll close the session for today.
Awesome. Thanks
Thank
all
you
for joining
all.
us.